bacterial transformation technique is a very important technique in microbiology and plant pathology laboratory. We use this technique to express some reporter genes. I guess all of you understand what is reporter gene. Reporter gene is the one which reports the microbial presence in an environment. Okay. Reporting. You can do this reporting by different methods, and this bacterial transformation based reporter gene expression helps you to visualize the bacteria directly using microscopic techniques. Okay. So, can you tell what is the most famous reporter gene which is used in microbiology? Fluorescent protein expressing genes. It may be green fluorescence or it may be red fluorescence, cyan, different variants have come of it. Okay? But among them, the most popular choice in most of the laboratories is green fluorescent protein. How many of you have prior experience on bacterial transformation? Okay, one hand is raising, another hand is raising. Right? So you might have studied in your lower classes. There are different methods by which you can transform microorganisms. The classical three routes: conjugation, transformation, transduction. These are some of the tools, some of the terminologies you might have read in your junior classes. The practical what we are doing today, bacterial transformation using a special apparatus, what is called as Electroporator. So Electroporator is a machine which creates the conditions favorable for transforming the bacteria by a mechanism what is called as direct uptake of free nucleic acids. Okay. So the classical definition is bacterial transformation. Okay. Parallelly, you might have studied one more method by which the transformation can be done. That is through conjugation. Okay. What you are practicing today can be done even by conjugation also. Right. So conjugation is slightly longer route and electroporation is shorter route. So we are not going to demonstrate conjugation because we need more days and more tools are needed here. So we will directly demonstrate whether you practice in each group this simple transformation technique. Okay. So before that, we must have certain idea about the whole system, what we are going to demonstrate. If you do transformation, what do you expect out of it? The reporter gene is going to express. The expression of reporter gene, if it has to happen, what should happen in the bacterial gene? What do you mean by transformation per se? It's a mere entry of DNA into the bacterial cytoplasm, or it is an integration of entered DNA in somewhere in the genome of bacteria. Okay, this is what we are going to demonstrate here. The vector system, what we are going to use today is TN7 based vector system. Okay, so TN7 based vector system is very unique. Unique in the sense, this vector precisely integrate the DNA in a specific genomic region in bacterial gene. Okay? The specific genomic region in the bacterial genome is popularly called as neutral site in the gene. This is very, very important. So if you integrate the reporter gene randomly, then there may be a possibility that the default traits may be altered. Okay? So that is what is called as random insertion of the nucleic acids. So that is not the one we are going to do here. We are going to do the precise integration of the nucleic acid, which is carrying the so-called reporter gene, in a neutral site in the genome, and this neutral site is constant across all gram-negative bacterial species. Okay, this neutral site is located in a genomic region that is next to a coding gene, what is called as GLS-M. This particular gene participates in the biosynthesis of N-acetyl glucosamine, an important cell wall component of most of the gram-negative bacteria. Right. The TN7 system recognizes the genomic region which is lying just next to GLS-M. This is the gene. GLS, all small letters, M capital. The GLS-M site is the one which is receiving this nucleic acid, which is containing the reporter gene. Okay, so we are going to use two vectors. One vector is carrying the reporter gene, and another vector is supporting the whole integration process. We term that particular second vector as helper vector. Okay, so there are two vectors are involved. One is a donor vector, another one is helper vector. In other terms, the donor plasmid and helper plasmid. Okay. So the donor plasmid is having the reporter gene. The reporter gene is a gene which is coding for green fluorescent protein. Okay. The same vector, there are a number of antibiotic resistant cassets are built in. Okay. One such antibiotic resistance cassette is a resistant cassette which is conferring resistance against gentamicin. Okay, so that means after integration, we expect that the genes or the reporter gene which is carried in the donor plasmid is stably expressing after integration in the neutral site. Okay, once it is expressed, the bacteria which got integrated, which received the nucleic acid along with the reporter gene and the resistant cassette, is going to grow in an antibiotic medium, in a medium which is containing antibiotic. In this particular case, it is gentamicin. Okay, so this is one side of the story. The second side of the story is we also need a helper plasmid. The helper plasmid, what we are going to use today, is having a resistance against an antibiotic, what is called as ampicillin. Okay, so there are two plasmids. Plasmid number one is a donor plasmid, which is carrying the gene for green fluorescent protein, and also a resistance conferring gene, a gentamicin resistance conferring gene. Right. Once it is integrated, the bacteria which receives these genes is going to grow in a medium which is containing antibiotic gentamicin. Okay. So what we have to do now? So we need to mix the target bacteria. Remember, the system what we are going to demonstrate today is a gram-negative bacteria. So any one of the gram-negative bacteria you can take. For this practical, so we have kept Pseudomonas foetida as a test organism. Right. This is one of the gram-negative bacteria. Right. So this bacteria is sensitive to the antibiotic gentamicin. Remember, this is one of the criteria. Whatever bacteria you are working on, suppose if you transform X bacteria, ensure that the X bacteria is sensitive to gentamicin if you want to use this particular system of transformation. Okay, so we have done a lot of prior analysis, prior testing, and we found that Pseudomonas foetida could not grow in a medium which is containing gentamicin, even at 20 microgram per m. So that is already established. So having established that, we have decided, okay, we will go for a TN7 vector, which is having resistance governing gene, that is gentamicin resistance governing gene for this transformation experiment. And we have a helper. The helper is having resistance against ampicillin. Right. So for electroporation based bacterial transformation, the prior requirement is you need to have the cells of your target bacteria, the cells of the target bacteria for which we have created a competence. In other terms, you have to keep the competent cells of the target bacteria. Okay? So you start from that experiment, it's a very small experiment. You need to confer this competence on the Pseudomonas foetida cells. Right. The competent cells preparation for this particular electroporation based genetic transformation is very simple. You take a mid-log phase culture. Mid-log phase culture means the culture which is in the active growth stage. You take the bacteria which is having a OD value of around 1 to 1.5 maximum, right? So then you just clean the cells. Cleaning the cells means you have to go for a centrifugation, right? And go for repeated washing. Okay, the washing is done with the help of sucrose, that is 300 millimolar concentration. So 3 to 4 times washing you do, right? And finally, whatever cells you harvested after 3 rounds of washing, you just suspend in 100 microliter of 300 millimolar sucrose. Okay, the competence cell preparation for electroporation is very, very simple. Right, you need an overnight culture, number one, and you need to spin down or you create a pellet. Right, the pelleting is done at very medium centrifugal force. You don't have to go for very high speed. We just use around 800 G force for about 2 minutes. Okay, so I'm sorry, 8000 G force for 2 minutes. Okay, so cells will be pelleted. Remember, you need a viable cells here. So unlike your DNA isolation protocols, you might have used a very high speed of around 12,000 RPM. So you don't have to go for such a very high speed here. You just go for a medium speed of around 8000 G force. So it depends upon the rotor. The speed varies. It may be around 5200 RPM. It depends upon the rotor. So the rotor what you are going to use today, it supports both. So what you do, you spin down and collect the cells and go for washing, 3 rounds of washing. Right. Each time it is again the same speed, 8000 G force for 2 minutes. Right. Then after the 3rd round of spinning down, you just suspend your bacterial cells in 100 microliter of 300 millimolar sucrose. Okay, your competence cell is ready. Right. Now next, what you have to do? In bacterial transformation, what happens? There's a free nucleic acid. The bacterial cell is in the most receptive stage. They interact, and somehow the DNA enters into the bacterial cytoplasm. Question is, what is that somehow? Right. So this somehow is created by Electroporator. Right? The Electroporator is a machine which generates very high electric voltage, often around 2.5 kilo volt, 2500 voltage. That is created. So this high voltage will give a shock to the bacterial cell. Right? So we expect the shocks will create non-lethal force on the bacterial cell wall. right, in a microsecond. Right? It is something like, it is not lethal at all. If it is lethal, this technique will not work. It is non-lethal. Okay? In a microsecond, it is shock. Shock means the pores are created on the cell. Right? And these pores are tiny enough to accept the foreign DNA into bacterial cytoplasm. Okay? So, you have to mix the competent cell with your donor plasmid and the helper plasmid. Okay? The donor plasmid in question today is plasmid PBK, mini TN7, GFP2, GF10. Okay, this mini TN7 is very important. It's a modular plasmid. Okay, somebody has constructed it for the purpose of delivering the foreign DNA in a specific neutral sites in the bacterial gene. Okay. The donor is PBK mini TN7, GM10, GFE2. Okay, that is the donor plasmid. And we have the helper plasmid. It's the PUX, correct?
Yes.
AMP100. Okay. So this is having ampicillin. This is going to confer ampicillin resistance. Okay. So the specific names are already given in the manual. Please write down. You can Google it out. You get more information about the vector map. You will come to know how many genes are there. Okay. What you have to do is you have to keep this donor plasmid and helper plasmid ready in your lab. Okay, before the infection. So that means, by and large, these vectors are maintained in a host bacteria. That host bacteria is E. coli. So this morning we had a lecture about E. coli databases. Okay. So the E. coli, what is being used to maintain these plasmids are XLI. So you don't have to handle E. coli here right now because we have grown the culture in a respective antibiotic.
तो बता देना उनको किससे ऐसे हो रहा है.
The plasmid is kept ready at 100 nanograms per microliter. This is very important. The question comes, how much of DNA which represent donor plasmid and the helper plasmid you need to mix with the competent cell. Okay, the competent cell you can mix 1 ml of So the competent cell you take around. I already told it is 100 microliter you are going to suspend cells. Right? The starting material is 1 ml. Right? So 1 ml of actively growing Electroporator culture is started with. You spin down 3 times washing you did 300 millimolar sucrose. Now finally you are going to suspend in 100 millimolar. Right? That means whatever you started with is going to be concentrated 10 times. Okay, so this 100 millimolar of your competent cell preparation will have anywhere between 10 to the power 9 to maybe 10 to the power 10 cells. That is very high dose of bacterial cells. So you take these cells, maybe 50 microliter you take, and mix it with the plasmid DNA, that is the helper plasmid and the donor plasmid. Both of them may be around 50 to 100 nanograms. Right? When you do this experiment, don't do it with one particular concentration. You put a range of concentration. Maybe starting from 50, 100, go up to 200 hours. Okay, that means 50 and 100, you are presenting almost double the number of plasmid molecules on the competent cells. Okay, so now you mix it thoroughly. Right? After mixing, you subject this mixture, a mixture of your competent cell, your donor plasmid, and the helper plasmid. Right? High voltage. The question is how to create a high voltage. Right? So you need to have a special apparatus which can allow the electric current to pass. Okay? And this is performed by a small gadget, what is called as Electroporator keywaves. Okay, this is unlike, it's something like, in spectrophotometer emitter used to keywaves, right, to take measurements. Here it is same, but it is having a special attachment in the bottom which allows the electric current to be passed. Okay, we will show you how it looks like, and it is slightly expensive, right? And you take that, and there are some conditions. You need to use ultra clean the EQs. We call it as EQs, right? Number one. Number two, you have to pre-chill it at least for a couple of hours, either in ice or in -20. Okay, the EQs are kept in ice or -20 for a couple of hours, right? So once you kept everything ready, you have the cells ready, you have the plasmids ready, you are about to mix everything, so at that time you take it out, right? Then you just load 50 microliter of your competent cells plus 50 nanograms of your donor plasmid, 50 nanograms of your helper plasmid. Okay, very gently you mix it. Okay, after mixing, you keep it in ice for about 15 minutes to 20 minutes. To just keep it in ice. Okay, the idea is you are enabling the kind of interaction between the cells and the nucleic acids that you present. Remember, 3 different kind of things are here. One is the competent cell, number 1. Number 2 is the donor plasmid. Number 3, helper plasmid. Okay, so after this 15 to 20 minutes, now you just load the whole mixture in the Electroporator QS or EQs. Okay, just present it inside. Now it's ready for electroporation. Okay, now we go to the machine. Right, this machine is available in our lab, and just present this, insert this EQS in a slot which is there in that machine. Then you follow the instructions that is given for this operation, the Electroporator. Okay, so then you record the parameters what the machine is going to give you. Mostly the quantum of voltage, number 1, and how much time the cells are exposed in terms of milliseconds. Okay, those data you just collected. So once the electricity is passed through this the mixture of cells and plasmids, you take it out, right, and quickly you recover that and put it in a media. And this media, I think many of you might have heard about it, a soft media. This was soft media. It's a special kind of highly nutritious media for bacteria to grow. Okay. Sometimes people use even LV media also. Okay. The idea is it is already given a shock at a higher electrical voltage. So what do? Put it in that new microfuge tube. Immediately you add this media. So that allows the bacteria to recover from the shock. Okay. So after mixing with the media, you leave it for about 5 minutes in the workbench. Okay. Then you allow them to grow for about 1 hour in an incubator. This is called outgrowth. Okay. You allow the bacteria to grow, recover first, followed by the growth. So both you have to do. Okay. Recovery is just by adding the soft media or LV media. Then you allow the bacteria to grow for about 2 hours. Alright? Okay? Now you expect that some of the cells or many of the cells had an opportunity to receive these two plasmids. There are numerous possibilities here. Cells might have received nothing. Some cells might have received only donor plasmid. Some might have received only helper plasmid. Some cells might have received both of them. Right? So all kind of possibilities. You just imagine and write it down. Okay? Now, you need to know whether the transformation really took place or not. Okay? What is the best way to know that? You allow them to grow in media which is having okay, so there is no need to add ampicillin here. So because the main vector is your GFP vector, which is having gentamicin, right? So you just add gentamicin here. That is good enough, right? So now after 2 hours, you take it out. Now how much you have? Each group you will be having 1 ml, correct? So 50 microliters recombinant cell plus both the plasmids. You added another 950 or even 1 ml you put, doesn't matter. It is around 1 ml of culture is with you now. It's got recovered, and also you allow them to grow for about 2 hours, 1 or 2 hours. This phase is what is called outgrowth. This outgrowth is very important. Okay, this outgrowth allows the plasmid, if at all it integrated, it entered, allow them to really, do the rest of the job. For example, recognizing the site, neutral site, integration there. Right, in 2 hours, at least a couple of generations, the cells would have undergone. So we expect in 2 generations, at least some cells, they got the plasmid nicely integrated in the neutral site. And now we have to bring out the colony which really got the plasmid integrated. This is called transconjugates. Okay, how to segregate the transconjugates from the wild type or those cells which did not receive the plasmid the way we want. Okay, the best method is you prepare the media which is recommended for this target organism. Right? In this case, it is LB we prepared. This LB is amended with gentamicin. Okay. Now we just spread on this gentamicin amended plate. Now the question is how to spread it. Right? So there are numerous opportunities. You place all of them together in one plate. Okay? Normally we don't do that because in biological experiments we need perturbations. Okay? The best way is, always I am telling, the best way is, if you have enough resources, this 1 ml, you take 100 microliter and put it in 10 plates. Okay, if you are short of resources, at least 200 microliters, 5 plates. Okay, your experimental design should be like that. Right, you have an opportunity to pick the colonies from 5 different plates. Right, suppose your transformation efficiency is so high, then what happens? So if you put it in one plate, the purpose of isolating a colony is defeated here. You are going to see the long of it. So that is not the one we expect. We need well isolated, healthy colonies are needed. Okay, so you put it in 5 plates. Today I think they are going to give you around 3 or 4 plates, whatever it is, just spread it. Okay, after spreading, right, this is a spread plate. Don't go for 4 plate. Okay? Spread plate. Okay, after spread plate technique, you just put it in the incubator and wait for 2 days. Right, after 2 days, you go and see that plate whether something is growing or not. Okay, and the most important point here is the controls. Right, this is experiment what I was doing. So what should be the control here? The control here is the bacteria without plasmid. Okay, number 1. Your downstream experiments. For that you need to do these 2 independent work. Number 1 is insert conformation. Okay, this insert conformation is usually done by polymerase chain reaction. Okay, so in polymerase chain reaction, you have to have a primer sets. Those primer sets are going to amplify the vector DNA. Okay, but in this case, you are checking for not only the plasmid, you are checking for plasmid at integrated state. Okay, so you need to have a primer sets. One primer set is from the bacterial gene, nothing to do with the vector. Okay, another primer is from the vector. Okay, if at all there is an integration, then only you are going to see the amplicon. Okay, if you use a primer set from plasmid, both, then there may be a possibility that the plasmid is there, but it is not integrated. You will get PCR positives. So you should not do such a PCR here. You have to do a PCR which exploits the primers which is designed from the bacterial genome, and also another primer which is designed from the integrated plasmid. Okay, so here we are using the two primers. The detailed experiments are there, and one primer is going to complement with the bacterial genome, that is GLSM. Okay, we have the primer set which is going to bind that particular gene. So I already told you the neutral site which is lying after GLSM is the one which is going to receive this plasmid. Okay, so you take GLSM specific primer, and also any one of the region in the vector you design another primer. So then you set up the PCR. Then if at all you are getting amplification, that means there is a integration happened in the bacterial gene. Okay, this is how to prove the bacterial integration. Now that is not sufficient, because you are ultimately what is the objective of this experiment? The objective is the expression of the reporter gene should happen. Then only the experiment is successful. You understand? Now how to prove that? This proof comes from direct visualization of your transconjugates. Okay, suppose you got around 10 colonies or 50 colonies, whatever number, you screen in a phased manner, maybe 20 colony you screen at a time, right? If out of 20 you just calculate how many of them are positive for PCR, the integrated state. Okay, having known that, now you shortlist maybe from that lot, you go for confocal laser scanning microscopy or fluorescent microscopy. Okay, so in fluorescent microscopy you just see whether the cells are giving you the expected level of fluorescence. Remember, another factor comes here, the level of fluorescence. So you need to have a population of transconjugates to select the one which is giving a brilliant, exceptionally brilliant fluorescence. Okay, so that colony is now shortlisted, or rather preserved for your future experiments. Okay, so this is the whole outline of this today's afternoon practical. We kept everything ready. Now each group will handle one electroporation keybase. Okay, so bacteria is already kept ready, plasmids are already ready. So what I would suggest is, out of 5 groups, maybe 2 groups can go with one particular concentration, another 2 groups go with maybe double the concentration of the plasmid. Okay, and the last group may be without plasmid, so that we can keep that last group as a check, very important, right? Then the rest all can have a 2 replication of 2 different concentrations. Is that fine? Okay, so 50 nanograms, 2 groups, group 1 and 2, group 3 and 4 you go for 100 nanograms. Okay, group 5 0 nanograms of plasmids. All right, so now let us do that. So finally we need to have 5 tubes. Those 5 tubes will get into 5 into 3, maybe 15 plates of LB plus gentamicin. Everything is same except the plasmid. Okay, all are same. There is no difference at all. Remember, you have to use only competent cells in the negative control. Don't go for your wild type and use it. No. Okay, you have to use the competent cells, because you are taking the stock from a single source. You are taking your liquids from single source. That's very, very important. Okay, so are you ready now? Good. Okay, start now. You have to calculate. I don't know. We'll teach you maybe when we are doing qPCR, you will understand all those things. The theoretical account, please read it. It confers the transposase enzyme which is needed for the integration of the main plasmid into the integration. Otherwise, what happens? You tell me. If transposase is no, it may integrate, but what happens? It may go out also if that gene is there. Okay, it's a suicidal plasmid. Okay. Yeah, the theoretical account we are not teaching here for paucity of time, so we can always teach that, but practical is only practical today. So you will be exposed to this practical, the how to do this experiment. And that is the purpose of objective of this experiment. Okay, please start your practical now. Yeah. Huh? One by one you can go.
Group 1 and 2 you come first.
Group 1 and 2. Please move to laminar flow, because this experiment you have to do in a laminar flow cabinet, because we need to maintain the sterility, aseptic environment, and all those things. Okay. It is described in page number 73. Please just read it, okay? All the details are there. Alright? So, suppose if you do not have. Because the promoter region which is needed for this GFP to explode is there in the bacterial. Which plasmid?
Competent cell is all ready now. Competent cell is ready. ಈಗ 3 ಮಿಕ್ಸ್ ಮಾಡ್ತೀವಿ ಯಾವುದರಲ್ಲಿ ಮಿಕ್ಸ್ ಮಾಡ್ತೀವಿ ECC tube ಎಷ್ಟು ECC tube
Reference is there. Always don't miss. Seriously, transformation itself is a liquid disease.
separate outside. These both are same.
For 4 months just to get one transformation. Morning and evening, almost continuously for 4 months. Usually I transfer. Sometimes it is you need to have a right plasmid. There is no prayer information or prayer success story. It is better not to do it. People are having limited time. Can't be sparing so much of time for this unexpected. You don't know actually. Yeah. So what you do now, these 3 groups is standing here. You can have a break now. Have tea and come back.
practical
Same method is there for example that we always request. If it is a bacillus, then we have another vector system. If it is another gram-negative vector, we have another vector system. So most of the time, TN10 system is recommended for bacillus. Yeah. Commercial mode is now many companies are manufacturing it and they supply us. ठीक है ना? We have not tested. have breakthrough. हाँ क्या हो गया? अच्छा बढ़िया उनका भी कंग्रेचुलेशन सर
अभी स्टॉप कर दें
गलती उठा लेता हूँ
चलो सर हाथ हटा लो ये वाला हाथ हटा लो इनको हो रहा है ये आप लोग लिख रहे हो मैं उस पर जा रहा हूँ
ट्रांसफॉर्मेशन में क्या होता है उसमें तीन मेथड है कंजुगेशन ट्रांसलेशन ट्रांसफॉर्मेशन कंजुगेशन अलग टाइप का होता है ट्रांसफॉर्मेशन क्या होता है इसमें न्यूक्लिक एसिड है ना ट्रांसफॉर्मेशन हो रहा है उसको ट्रेस आउट करने के लिए GFP
आप
केमिस्ट्री
हाँ वो सर वही समझा रहा था कि कॉम्पिटेंसी का फर्क आ रहा है कि
Sir told something that for gram-positive bacteria, so we can use for TN10 vector. Which vector you are using? No, not TN7. Sir told 10. Sir told TN10. Okay. Yeah, I think
ये यूज हो रहा है they are not doing it here.
because both bacteria and gram-positive bacteria, gram-negative bacteria, their genomic structures, are different. No? Sir told that their phylogenetic analysis for this gram-positive bacteria different. Therefore, gram-negative bacteria, this phylogenetic tree is different. This positive phylogenetic tree grows this way, and this negative this way. Completely different. Are you using this tri-parental matching? But are you using both the plasmid and genes? Ralstonia solanaceae, RIP50 recipient strain. Okay. But do you use both this plasmid and Have you done it in this experiment? Okay. So you are using this one, no? The electroporation method.
Okay, then that seed will be kept for keep for germination. Then after germination, I will see that endocytes where it will reach. If to the posterior fluid or this stage of the stem, plant parts को cut करके फिर then the from plant again I have to observe this GFP stage.
Only GFP you are using, not another one. Other than GFP, red fluorescent protein. Sir told 3, I think, GFP, RFP, and then another one.
So many different.
Here, the term genetics was coined by T.H. Roderick. I saw that just at first examination, first evaluation, I just simply just overlooked this term. When I came to know something Roderick, I wrote Frederick. But there should be no Roderick. A geneticist in 1986 at the Jackson Laboratories. But you are using this method, electroporation method. समझ गया
from the different zones again. पहले आइसोलेट करूँगा आइडेंटिफाई करूँगा थ्रू सीक्वेंसिंग लेके फिर करेंगे इसमें फंक्शंस उसके बाद सेलेक्टेड जो which shows best to be को टैग करके सेलेक्टेड ऑब्जेक्ट्स पे एक 8 लेवल उसको फिर पिक्चर का यहाँ तक ये हो ये
How many gram positive bacteria isolated? Okay. Where from you see or collected?
6 zones of India. So 6 zones means one zone
from crop, no?
From seeds.
Seeds, okay.
Collected seeds from 6 zones.
From the seeds only you are isolating bacteria.
Bacteria and fungus.
Okay.
Mainly bacteria. Focusing on bacteria. Second from the compounds. Seed
How you are going to identify bacteria inside seeds? Sterilization. Seed could remove. Okay. But after removing, after sterilization, you are removing seed coat. But suppose bacteria are there on seed coat or just below of the seed coat.
Bacteria can be seed
bone enterprise. Okay, inside the embryo, do you get bacteria embryo?
क्या ग्रुप बना रखे हैं लोग जो बढ़िया दिखेगा उसी पर रहेगा दिखेगा उसी पर रहेगा नहीं मैं तो कर रहा हूँ ग्रुप क्या कर रहा है
forward reverse ಕಾಂಟಿಕ್ಸ್
ಕಾಂಟಿಗೆ ಯಾಕೆ ಮಾಡೋದು ಅಂದರೆ ಸೊ ಅದು
ತುಂಬ ಕಡಿಮೆ ಇತ್ತು. ಟಾರ್ಗೆಟ್ ಲೆಂತ್ ನಾವು ಕಾಸ್ಟ್ ಮಾಡಿದ್ವಿ NCBIಗೆ, ವಿ ಆರ್ ಗೆಟಿಂಗ್ 80% or 75% ಸಿಮಿಲಾರಿಟಿ ಇರೋ ಸೀಕ್ವೆನ್ಸ್ ತೋರಿಸುತ್ತೆ. ಸೊ ದಟ್ ವಿ ವಾಂಟ್ ಟು ನೋ ಎಕ್ಸಾಕ್ಟ್ ಸೀಕ್ವೆನ್ಸ್ ಎರರ್ಸ್ನ ನಾವು ರಿಮೂವ್ ಮಾಡ್ತೀವಿ. ಸೊ ಜಸ್ಟ್ ವಾಟ್ ಕಾರ್ಟಿಂಗ್ ನಾವು ಕ್ಯುರೇಷನ್ ಮಾಡಿದ್ದೀವಿ ಮಲ್ಟಿಪಲ್ ಕ್ಯುರೇಷನಲ್ಲಿ CLC ಸೀಕ್ವೆನ್ಸ್ ಅಲ್ಲಿ ನಾವು ಕ್ಯುರೇಷನ್ ಮಾಡೋದು. ಯಾಕೆ ಅಂದರೆ ಸಮಟೈಮ್ಸ್ ವಿ ವಾಂಟ್ ಟು ಕನ್ಫರ್ಮ್ ದಟ್ ಆ ಸೀಕ್ವೆನ್ಸ್ ಸರಿ ಇದೆಯಾ ಅಂತ ನಾವು ರೆಫರೆನ್ಸ್ ಜಿನಮ್ನ ಒಂದು 5-6 ಡೌನ್ಲೋಡ್ ಮಾಡಿಕೊಂಡು ಅಲ್ಲಿ ಏನು ಮಾಡ್ತೀವಿ ಅಂದರೆ ರೆಫರೆನ್ಸ್ ಜಿನಮಲ್ಲಿ ಕೆಲವು ಗ್ಯಾಪ್ಸ್ ಇರುತ್ತೆ, ಆ ಗ್ಯಾಪ್ಸ್ಗಳನ್ನ ಇದು ರಿಮೂವ್ ಮಾಡ್ತೀವಿ. ಸೊ ದೀಸ್ ಆರ್ ಆಲ್ ಸೀಕ್ವೆನ್ಸಿಂಗ್ ಇರೋದು. ಸಮಟೈಮ್ಸ್ ಬೇಸ್ ಕಾಲ್ಸ್
टाइट नहीं टाइट नहीं था भैया प्लीज, टाइट नहीं था, नहीं हो गया ना। हाँ
ಅಲ್ಲಿಗೆ ಏನಾಗ್ತದೆ ನಾವು ಇನ್ನೊಂದ್ಸಲ
I think some of you can come here. We'll just explain this one. Yeah. ठीक है. Okay, just form a semicircle. I think you can come here. You can come here. Be responsive, please.
Yes,
I was talking about the machine which is helping you to deliver the DNA into the bacterial cell. This is that machine. It's called Electroporator. And the brand name here is E-Porator from a company Eppendorf. Okay. If you connect it, it will light up. And this is the most important one, what is called as the slot to keep your EQs. Okay, so you just insert it here, then just put it back inside. Okay, then there are 2 programs by default. It is available in this machine, and it depends upon your requirements. Usually we go for 2.5 kilovolt, right? For about 4 to 5 milliseconds. Okay, you just load it, just start the machine. It's very simple. Don't think that it is a very complicated experiment, not at all. Right, just keep the EQS inside, then insert and press this one, then it will give you a small noise for about less than a second. Okay, then it will display the readings. The reading, how much voltage was allowed inside. Number 2, how much time. Okay, that is called as time constant. So you make a table. Okay, sample 1, how much voltage and time constant. So make a table, and this time constant is so important. Okay, the most successful transformation happens in a minimal, the time point. Time constant, 4 to 5 milliseconds. Okay, and take it out and remove the QS, bring to flow cabinet, then you add the sock medium or LV medium directly into this QA, then just mix it, repeat it, what is called as up and down, right, very gently, right. Then you transfer to microfuge tube, then allow it to grow for another couple of hours. Okay, then after that you take it out and just plate it on antibiotic amended medium. All right, so how many of you have seen this machine before? Yeah, those who have seen it, not seen, surprising, not seen it. Okay, so different companies are manufacturing this, right. So this is one of the famous model of Electroporator, and the Electroporators are available for prokaryotic system, and you have Electroporator for eukaryotic system also.
they have another model that is Multiporator, that is available both for prokaryotic cells and eukaryotic cells also. Okay. So this is an optional. Please keep in your mind, without this machine also you can do the transformation. If you are not having this machine, it does not mean that you cannot do transformation. You can do transformation. That is through conjugation route. Right? Conjugation is an event wherein you mix different bacterial cells and allow them to exchange the genetic material. Okay? The method what is used in the same experiment, if you do not have Electroporator, then you can do this tri-parental meeting. Okay? So I was talking about ampicillin in the helper plasmid. There it play an important role. Okay? So what we do, you take the target bacteria, right, and you mix it with a host, E. coli host, which is carrying this donor plasmid, and another E. coli host which is carrying the helper plasmid. You have now 3 bacterial setup in your laboratory. Your target bacteria, it may be Pseudomonas foetida, Pseudomonas fluorescens, right? So then you have E. coli, which is having this plasmid, the reporter gene containing plasmid or the donor plasmid, and you have your helper plasmid containing E. coli. Now what you have to do, you grow all of them the previous day, grow overnight. Okay, grow overnight, and you have the overnight growth of all the 3 bacteria. All right. So the following day you take 1 ml of these cultures, right? You just spin down, remove the media, resuspend in a plain media, maybe a plain LB broth, right? Then mix all the 3 together in a different ratio. Okay. So here also we did the same thing. You took 50 microliter of competent cell, you mixed with 50 nanograms of helper and 50 nanograms of donor. Same way there also, you have 1 ml of your target bacteria, you put 500 microliter of your helper and 500 microliter of your donor E. coli cells. Okay, mix all of them together, right? Then the mix of all the 3 bacteria, you take a plain LB agar and put nitrocellulose membrane, right? On top of that you just load the mixture of all the 3 bacteria, right? Grow them overnight, and this overnight is just to enable the bacteria to interact with each other through a process what is called as conjugation. Okay. Now all the 3 bacteria, they conjugate, and what is needed for you is your target bacteria which received a helper plasmid and the donor plasmid from the respective E. coli host. The trans conjugates are needed here. Okay. So then you take the mixture of all the 3 bacteria and just plate them in a media which is amended with different antibiotics. Okay. The one which is needed for you here is your target which received the helper from one E. coli and donor from another E. coli, right? So in a mix you have so many other things which you don't want. For example, you do not want E. coli at all. You do not want E. coli 2 also, okay, which is having the helper one, and you do not want wild type which never interacted with the other 2, right? So you have to remove all of them. So how will you remove all of them? Antibiotic. So here comes the trick. So before setting up your conjugation experiment, you have to do a small laboratory experiment. The laboratory experiment is, you have to select your target bacteria in antibiotics which is having the resistance is controlled by chromosomal borne genes. Okay, we have some antibiotics, the resistance is completely governed by chromosomal borne genes. For example, rifamycin or rifampicin, the rifampicin resistance is built in most of the prokaryotic cells. The only thing is the frequency varies. Okay, maybe you will get 1 or 2 cells in a population of 10 to the power 9 or even 10 to the power 10 cells will have the rifampicin resistance. So what you have to do before designing this conjugation experiment, a single day experiment, you just plate out your target bacteria in a plate which is amended with rifampicin, and you put a heavy load of bacterial cell there, maybe 10 to the power 10 or 10 to the power 9 cells, you just plate it. So you just put the mutation frequency, you get 1 or 2 colony which is appearing, and you pick those 1 or 2 colonies. Remember, these 1 or 2 colonies are not mutants actually, right? They are native population only, right? Otherwise I will put it like this. This is intrinsic antibiotic resistance. The cells which is having intrinsic antibiotic resistance. Okay, you select the one cell or couple of cells which is having which displayed the rifampicin resistance. You take them on board. You confirm once again by inoculating in a broth which is amended with the rifampicin. You got it? So you see that the growth is happening much like the wild type which is growing in no antibiotic broth. Okay, that means you have a cell or a derivative of your wild type which is having rifampicin resistance. Now this derivative, all the cells will be displaying rifampicin resistance, unlike the parent which is having only few population or 1 or 2 cells. Okay, this material become an important material for you. Remember, this rifampicin resistance is not altering any other bacterial traits. All other bacterial traits are intact here. So none of the traits are altered. If it is a plant pathogen, it is still a virulent pathogen causing disease. If it is antibiotic producing bacteria, still it is producing antibiotic. If it is some enzyme producing bacteria, it is still producing enzymes. All other characters are intact. Right. The only thing happened here is you selected the cell which is having rifampicin resistance. Now that becomes your target. This target you are mixing with other 2 E. coli. Right. When you want to select the transconjugants, so what you have to do is you take an antibiotic plate where you add rifampicin and gentamicin. Okay, rifampicin and gentamicin. So gentamicin will allow the transconjugants to grow. Rifampicin will not allow the E. coli to grow. Okay, so this is what is called how to counter select the E. coli in a triparental mating experiments. This is how it is done. So in the selection plate, you have a rifampicin and also a gentamicin. So what happens? What is growing there? They are transconjugates. Okay, that conjugates are selected. Again, it is subjected to the 2 other experiments which we discussed this afternoon. One is PCR for integration and another one for the expression of the GFP protein in confocal microscopy or fluorescent microscopy. Okay, is that clear to all of you? Right. So what I mean to say here is this machine is a luxury. This is not really needed. Okay, if it is there, you can do this experiment in a matter of 2 days. Otherwise, it takes 3 days. That's it. Okay, and then you don't need rifampicin at all here when you use this machine. Right, you just put gentamicin. That's enough. Okay, so and sometimes you get a good frequency of transconjugates in this machine, and this is high throughput. You can do transformation for several strains, right? And these are all some of the advantages with this Electroporator. Okay, now you proceed. We have limited time today, so you want to say something? Please come here.
Group 3, 4, 5 are they assembled.
Okay.
Experiment is going on.
Okay. Right. So you can do it here now.
Electroporation we will do it here.
Yeah.
But laminar work we have to do that.
Okay, competent cell preparation. So now the competent cell preparation is going on now. So plasmids are kept ready now?
Plasmids are ready.
Okay, so when are we going to do it? Right now?
Maybe it will take another 15 minutes.
15 minutes, okay. After 15 minutes, let us assemble here. Then for each group, we will do this electroporation right here. Okay. Then we allow for outgrowth. So during that time we'll break and complete the presentation, and then come back here and plate it out today and go. Is that fine with all of you?
Yes.
Okay, good. So other groups join Kuleshwar because he is demonstrating the competent cell preparation. At the time of experiment, each group will go to that machine. They will do. So the 5 people will explain. On the board, no? It's not happening. It's not working. Any doubts for you? You are not understanding? What I explained at the start of this practical is Electroporator. You don't need E. coli at all. Okay, but if you do not have this machine, still you can do this experiment through a classical method, what is called as conjugation. Is that clear? Yeah. In conjugation, what happens? Bacteria exchange the genetic material through conjugation tube. Okay. So you are all doing this bacterial transformation in your thesis project? Not yet. Okay.
So we have never practical the using
Okay. Chemical method. That is also a nice method, no doubt.
Even if you don't do electroporation, you can do that also.
Chemical method also you can do.
Chemical competence. Arjun.
And you can transform fungi also. So in our lab we have done number of times. Slightly different protocol. We use a different vector system, eukaryotic vector system, not TN7 based. We use a P-Cambia based vectors, which is having the tDNA right and left border. We have vector system available, that is P-Cambia GFP. So this vector system allows you to, of course, it's not a site-specific integration; it's a random integration. But the tricky part here is you have to select among the transformants the one which did not lose any of its native traits. Okay, so that is the only trick. So what you do, you have a population of transformants and screen all of them for the intended trait. For example, if it is a plant pathogen, and ensure that after transformation it is still retained its ability to cause disease or virulence, whatever it is, then you shortlist them for future studies. So I think one of the PhD students, Asha Jyoti, she transformed one of the Magnopothy strains using GFP, P-Cambia. And it is unlike a 2 days work, that takes at least a couple of weeks. You need to go for a lot of planning, right? You need to create different vector system, then the Magnopothy, you have to grow at a specific concentration of cells, spores. Then you have to go for again filter meeting experiment only. So it works, right? End of the day, we make agrobacterium temifaciens, right? There is a famous strain LBA4404. That strain is mixed with Magnopothy. Then actually what's happening there is the agrobacterium temifaciens send out this P-Cambia GFP vector into Magnopothy. It's an infection only, right? Yeah. Since there is no virulence genes are there to cause a tumor, so Magnopothy only receives the GFP gene with antibiotic kanamycin and other antibiotic resistance. So it works nicely, and it gives an exceptional fluorescence. So what I am telling is the gene is same, GFP is same, whether it is a TN7 based vector system or it is P-Cambia based vector system or any other vector system what now commercially available, the gene is same. Okay, the people have created a kind of modular plasmids based on their needs. Okay, somebody created it for bacteria gram negative, somebody created it for fungi like Magnopothy filamentous fungi, and somebody has created it for even gram positive bacteria also. Okay, this is how the whole, but it is a very valuable tool, no doubt. Okay, once you do it, ensure that any transformation method, a single trial will not work. If it works, then it is you are exceptionally lucky. Right, you have to go for multiple trials. Okay, so then second, third trial you will be getting certainly the results. There is no doubt. That has been our experience so far. Okay, the only bacteria which troubled us is Pseudomonas aeruginosa. That only troubled us because we spent 4 months of continuous effort. Morning one transformation, afternoon one transformation. We won't get any results that easily. So end of the day, after 3-4 months of trial, we got one particular event. We could get around 100 plus colonies. All of them are fluorescing. So sometimes this kind of uncertainties will come, but it all depends how far you want to do it. So and how much time you can afford. So that matters a lot. Okay. The reason is some of the resident plasmids, in some bacteria, the incompatibility group plasmids, that will play a spoil sport. That will play a spoil sport. So your vector system and the resident plasmid, they may not go hand in hand. Then you have to silence them. You have to play a little bit of trick. All those things. Okay. So one of the easiest way to silence those kind of restriction modification system is prior to mixing, we normally subject the cells to 42 degrees Celsius for about 30 seconds. So at that time you just mix it with E. coli in conjugation in particular. So sometimes this trick works, right? Elevate the temperature to around 42 degrees Celsius for about few seconds, maybe 30 seconds, 40 seconds, right? Just take it out, then you mix it with the donor and the so-called helper. Then you proceed with outgrowth. Then just plate it out. It works. All right. Okay. Any more clarifications on this? Will you be able to do independently this transformation now?
For gram-positive bacteria, the voltage.
No, see, technically every organism you can transform, no doubt, right? But the relative, the ease, it varies. some bacterial system it's very easy. For example, Pseudomonas foetida, students, my research fellows, they could do it in a matter of 2 days, 3 days. The results has come without any problems. I already shared with you Pseudomonas aeruginosa. It took 4 months of continuous effort, right? Just imagine how much of transformation events we carried out continuously, non-stop for 3-4 months. So it all depends, right? For gram-positive bacteria, what you are asking, it has been always a challenge, right, to transform gram-positive bacteria. Right? Nowadays, vector systems have been developed. If I remember correctly, TN, because we no longer do that. One of my students did it. I think it's TN10 based transformation system, and again you have to remove the cell wall. It is always the protoplast transformation. You regenerate the cells after transformation of those protoplast. So that's a pretty long route, but transformation happens. It happens. Okay, it takes time. I told Magnaporth it takes 15 days minimum, right? No, total experiment time, duration, from the start to end. And you have to follow this experiment very, very religiously and on time. You cannot, if you need to be there on that particular incubation, expiry of that incubation, you need to be there. A little bit of follow-up is needed. Then it takes 15 days, the transformation. Right? A lot of interconnected events are there that you need to follow very systematically. Okay. So for gram-positive, resources are there nowadays. You can contact, just find out who is expert in that particular subject. And for Bacillus, there is another group working in Germany. I think if I am not wrong, Rebecca. Right? Her group is working on Bacillus megaterium. So they have developed a lot of transformation system for megaterium. Okay, Bacillus megaterium. I think most of the vectors what they developed is for transforming Bacillus, and we were in the sense we received a construct from those labs as a gift, and we did it here, and we got successes. Okay. So it is there for any organism you can transform. There is no problems on that. Okay. Only thing is how far you are following it up. That's more important. If something else, some other academic activity comes and you are diverted, then nobody can help. That's the issue. Okay. So you need to continue the experiments. Whatever experiments you are doing, you need to. There must be a logical conclusion. Okay. You start it and halfway through, if you give it up, then it is difficult. Okay, you need to give a proper follow.
Keep trying.
Yeah. If first attempt fails, second attempt, because you learned in the first attempt.
No, you can able to come across where you are committing a mistake, which step you are committing mistake, where you can able to manipulate in the sense modify the steps. Okay, like that now you can troubleshoot where you can do. But doing the troubleshooting, you need to understand the things, protocols very clearly. Which step is doing what. Okay, what is the mechanism in each step? So if you are knowing that one, then you can able to easily modify the protocol.
Yeah,
what are told at the trial, everything will come by trial and error. Okay, you can able to make your own SOPs.
Exactly. The key learning, especially fungal transformation. Unlike bacterial transformation, in fungal transformation, we know the fundamental difference between fungi and bacteria. In a plate, how many colonies of bacteria you can accommodate? 150? 300 colonies? Easily you can accommodate. Can you accommodate 300 fungal colonies? Not possible. Nah? So this we know it already. But when you do a fungal transformation, this is one of the most important points. Right? Since our group, we are having expertise in bacterial transformation, we always believe we need 10 to the power 9 to 10 to the power 10 cells. Right, so that the probability of bacteria interacting with the vector DNA, we give maximum opportunity. Right, but again, you need to have optimum dose of bacterial cell and plasmid that is there. That comes with the experience. What is the right combination of cells and plasmid DNA concentration for satisfied the electroporation that need to come with experience. We simply applied this principle in fungi, utterly failed, right? It should be the other way around. You should not aim for more number of conidia for fungal transformation, as minimum as possible, right? Same effort, everything same, only one difference. The difference is, instead of 10 to the power 8 conidia, we have taken only 10 to the power 2 conidia. That means just 100 conidia in 1 mm. Right? Out of 100 conidia, 21 of them got transformed. You understand? So, what happens is, if you put too many target cells, then what is called as a star activity comes, false positive. It is not actually transformed, but they grow in antibiotic amended medium. Right? They are not transformed one. Right? So, you need minimal number of cells, 50 to 100 cells. You transform, you will get a good result. So, the point here is, this optimization you will not find in research papers. That need to be done. There is no other option. You have to do it. You have to customize for your setup. Whatever organism you are handling, you have to make it working in your setup. That is more important. That is what is called as a trial and error. It is always trial and error, isn't it? It's always trial and error. So error is expected all the time. So let us see.
Commit the release, and only you can get the support.
Okay, good. So let us hope some of you do it independently in your thesis project. Isn't it? You have this in your thesis project? No. What's your thesis project? Okay, good. Okay. So V3, V4 or culturable microbiome. Okay. NGS? MISAC you are doing or cultured one? Uncultured one also. Okay. Only uncultured. Okay. Good. Anybody this side is doing this? Decided. Okay.
He is MSc first year.
In lab we are planning to use GFP to see whether our hierarchical archaea can act as endopermeable, which we are using.
Okay. Archaeal transformation I know, don't have any experience, but I don't know. Read a literature. What is the organism? Allotric, something like that? What organism you are handling?
I am handling hierarchical as well.
Okay. Gram negative. Archaea.
I don't know anything about it. So it's just now.
Isolated already.
It is previously isolated characteristics. That work is going on from last 10-12 years.
10-12 years. Oh, that's great.
Continuation of that.
There was one NASL project.
Acha.
Archaea.
Okay. Exthermophiles.
Extremophiles.
Okay. So what's going on in the other group? Other 3 groups? When are we going to? Okay. Good. So what is Divya working on now?
So genome-based analysis of rice actinobacterial interactions.
Oh, right. Okay. You made a presentation also. Okay. Actinobacteria.
Yes.
Okay.
This is in the preliminary screening also.
Okay.
Started the 4 objectives.
Okay.
So in that case, I just have a doubt. So today morning, sir talked about the databases and all that. He said before ending up with any genomics tool, we just check out whether the same data is available in the database, otherwise we end up with the loss of time.
Duplication of efforts, yeah.
So if we have so many isolates with us, we are thinking it is a new one and we are isolating. By the time if it is the database, someone has uploaded the same isolate what I have, the genomic data and all that. So what I'm supposed to
Somebody else will never upload the isolate what you have, unless and until you share the isolate with them.
So the isolate which you have isolated is your isolate.
Okay.
So remember, if the population is a non-clonal, then what species you are handling will be completely different from somebody else's species. So most of the time we believe the species what I handle is handled by X and Y in another country or another lab. That is not true. You isolated a bacterial species, you identified the species. Now your isolate is your isolate.
Okay, I say, but
you give a name based on the origin, host origin, or your name, whatever it is. That will not be available in somebody else's lab. It's not possible unless and until you share the culture. So that means every isolate is distinct and unique.
So then, what context he actually referred, like the repetition should?
What he told is there is a point also. Many times, if two isolates, for example, you leave now, after a few years somebody else joined, right? And the somebody else also got into the same program what you are doing, in particular the genome sequencing, right? Then it is a waste of time. Okay, same isolate need not be sequenced 2 times, not necessary.
So now
Right, if it is another isolate, then yes, there is a reason, because isolate variations are always there. Okay, provided the population is non-clonal. If it is a clonal population, then again you have to put a timeline, right? In a short succession time, if you are entering into the same rice field, whether she isolated some bacteria, you also isolate in a matter of couple of weeks, and both of you isolate the same. You do not know each other. Independently you do, then the chances of duplication of effort is there, right? So what I mean to say is, in my opinion, maybe with the passage of time, the falling cost of sequencing efforts will force us to do whole genome sequencing of every organism. That is going to be the reality, okay, with the passage of time. So now already it is sequencing is not a expensive experiment nowadays, right? So once upon a time it was in a few labs. Now even 50 MB genome, now people are doing in a small group, right? In an individual principal investigator level, the sequencing has come, right? That means what? So every passing day, a new technology that is cheaper than the earlier technology is coming to the literature. Affordable also. So better bioinformatic tools, pipelines. Now we will be able to do the genome assembly in a matter of couple of days. That's what Dushyant was highlighting other day. So what I am telling is, morning speaker also told, single gene sequence based identification is slowly fading away from literature. Now we depend more and more on multiple genes. That's what I was highlighting in my talks. 7 loci based identification is there. That will finally, this MLST also will fade away. Now that will be taken over by the WGS only. We will be in a stage after a few years, maybe a decade from now, every organism can be whole genome sequenced. And you have the pipeline in your own laboratory, your computing facility will be much better than what we are practicing now. And you are in a position to identify the organism by comparing the whole genome rather than a fraction of or one among the 6,000 genes which is there in bacterial genome. Many times that argument is there. You are identifying the organism based on only one gene, and how reliable it is. That question is always there. Okay, so that and all will go slowly, and will make, now it's a multiple gene. Now the policy is you take one more marker gene, and if you are handling 1 or 2 organisms, you can go for whole genome sequencing. If you are handling hundreds of organisms, then the question of time and cost arrives. Then only we deploy the tools like multi-locus sequence typing, population microbial population genetic analysis. So now it is one of the ruling technologies wherein you can use number of isolates in affordable cost in your laboratory, because just imagine sequencing cost for 7 loci is definitely far cheaper than sequencing cost of the whole genome of 100 isolates.
In that case, you can take only the potential or representative isolates for WGS.
That's what I am telling. See, it all depends. So nowadays, always multiple genomes. Recently, there was a paper in Scientific Reports last year. They have sequenced 100 Xanthomonas oryzae, Petavar oryzae. 100 isolates, whole genome. Of course, it's a resequencing, but nevertheless, it's a whole genome. So you see the amount of money involved, amount of time involved, right? It is, if it is affordable and you find a reason for doing that work, or you have a biological question which that will be addressed only when you do this 100 genome, then nobody can stop you. You argue that yes, I need to do this to address this biological question. Then you do it. Nobody can question your interest. Okay, and ultimately you have to decide what genomic tool is the right tool for your setup, whether it is WGS or it is multi-locus sequence typing, or even single gene based characterization. For example, microbiome. Whoever is doing microbiome for prokaryotes, we use only a fraction of the genome, that is V3, V4. That is only 400 base pair in length, right? So we believe that is having the species signature, and with that I will be able to come out with the so-called beta diversity, alpha diversity, and identity of those reads. Then it is fine, because end of the day your work and your data should be accepted by the peer reviewing. So that you need to keep it in your mind all the time. Okay. So of course the duplication of effort need to be reduced. Otherwise you will be unnecessarily wasting your time and money. That you need to keep in your mind all the time. Okay, na? Vinay's work, I already told you, you think of doing whole genome sequencing of your trichoderma, that carries some weightage rather than re-identifying the trichoderma, which is already identified. It's of no relevance. You tell your advisory committee you need to do that and try to seek funding for your whole genome. Then there is a strong logic, isn't it? Only one trichoderma you can do it. So other programs are yet to be finalized, correct? You are all MSc students, correct? Or first year? Okay.
So with the available WBS data, we can ourselves, one of your suggestions during class presentation, we can only design an additional marker for the
you can design. Yeah. If genome is there, you can design. You take some genes which is other than 16. That is recommended now. You need to go for one additional marker. And always it is polyphasic. You cannot depend on sequence based identification all the time. It is always polyphasic. You have to go for maybe metabolic fingerprinting or what is called as phenotypic fingerprinting, but Biolog, there is a company, or you can go for FAME. And last month we were in Pune. They have a multi-TOF based bacterial identification now. So you can, it all depends. Right. Many times you make a compromise. You identify just because you got matching sequence in GenBank or any other database. Right. Then you make a compromise. Okay, it is already there. I have identified. Suppose if that is wrong, then you are losing the credit. It may be a completely new bacteria. So nobody, very few students, they enter into this domain, characterizing completely new bacteria by DNA hybridization and claiming for new species identity. So that is a real contribution for science, especially in microbiology and plant pathology. So most of the time we just compromise. Okay, I have identified my species because it is matching with so and so species reported from so and so country. So that's not a good statement, right? You will be matching your sequence with somebody published from other country, maybe China or Japan or US or so. How can you say that just by having a match? You can only say that my strain is having closest similarity with so and so. That's it. You cannot say that you have identified your species. You only say that my strain is having closest match with so and so at 1400 base pair length at 97% similarity index. That's it. Let the peer group to form opinion about your data, isn't it? So unless you are confident and you are about the identity, you cannot wrongly name your isolate. right? Otherwise you will be wrongly naming your isolate and so and so. That is not acceptable. Okay. Okay, good. So culture collection, cultures are safe in everybody's hand, or you are mishandling the cultures? Many times now you start with something and you end up in something else. Isn't it?
Yes, it happened with me with LSC.
You start with something and you end up in something else.
So partly we are doing the preservation of the cultures of the stocks.
How do you preserve it?
Glycerol stocks.
Okay.
Enough or something else to be done.
Glycerol stock in what temperature?
So that coming to minus 18.
That is not to be touched at all, you know that? That is not for a routine isolation, no?
No, that is a permanent stock.
That's a permanent stock. If you lost all options, then only you have to go to minus 80. Minus 80 cultures are not to be taken out.
No, so that's what a permanent one backup.
You should
Yeah.
have a backup.
What is the regular culture you have in your lab? I am asking. Working culture. How do you maintain the working culture?
So in Naga we have slants or plates.
You are comfortable with that?
Till
Then you will end up like this. You started with something. Serial transfer, you are telling. Serial transfer. No, that's what I am telling you. Serial transfer. Yeah, short term because. Yeah. So 3 months. Then after 3 months, you will put it in another slant. Nah.
Regular work I need to
for
do that.
bacteria, fungal dispose it's okay, so it will be viable for long time. So in case of bacteria, what would be the time?
So we have techniques. We have techniques. Short term preservation.
You are having a bacteria which is producing spores.
Right. So always remember, you preserve the culture. I already told in the first day, one colony, a portion of the colony goes for identification through sequencing approach. Okay. The other portion goes for preservation. The other portion, half of the tips will go to minus 80 and the rest will go to minus 20. The minus 20 culture is a short term culture. Okay, so all are one only now. You are not picking another colony. Right, and you are not going to do repeated subculturing on that. No need. Minus 80 culture never touch it. Put it in a proper place with a proper location identity. Minus 20 culture, you handle on a daily basis or whatever, whenever you want. Take it in a mini cooler and take it to your flow cabinet. Take little bit, streak it and put back inside in minus 20. Minus 20 you can, because it will be still in the liquid state only. Okay, in minus 80 it is in the frozen state.
Daily routine use we can.
Minus 20 you can use comfortably, there is no problems on that. Okay, we in our group we don't keep the slant at all. There is no slant concept in our group. We don't use slant, we don't use cotton plugs, nothing. Because the stock is same. Yeah, the only risk here is the stock should not be contaminated. That's the only risk. That you need to put appropriate marker system.
If you are going to utilize that slant in within some short time, then you can keep it in. Otherwise, better than that, you can keep it in a minus 20.
And it is always better, the moment you identify the organism, you immediately develop a glycerol stock of reformation resistant colony. Okay. So because I already told you some time back, when you select a reformation resistant colony, it retained all its original traits. But you found another system of recovering the cell by putting reformation. Okay, what happens is, if you put reformation 50 or 100, it's very the probability of getting a contamination is very rare. Okay, because if you put the mutation frequency or the resistant type frequency is very low, right? So if you forcefully develop a strain which is having reformation resistant, all the members of that colony is resistant to reformation, right? So you put a reformation intrinsic reformation resistance as a kind of marker system, right, and make a colonies, and one set will go to minus 80, another will go to minus 20. This minus 21, every time you use reformation as a marker, antibiotic resistant marker, you streak it, chances of making another colony is very rare. So that should be the strategy. Okay. So by the way, rifamycin is light sensitive and not soluble in water. Okay, it is soluble in DMSO. So you dissolve in DMSO and protect it from light and use it. Okay. So likewise, you can the original, the biological purity and genetic purity also, both are addressed here. Okay, genetic purity is addressed because you already handled a rifamycin tagged, rifamycin resistant tagged strain. So genetically pure, and by adding antibiotics you can avoid all other contaminations. Okay, in a single stroke you are addressing two important in the sense issues. One is biological purity, another one is genetic purity. It's very, very important. It's more so for the strain which is elevated to whole genome sequencing, right? The whole genome sequencing, the culture has to be preserved. I already told you, culture has to be preserved in the original format. Okay, if you subject them into subculturing cycles, the chances are that they may lose its originality. Don't say that it may lose its viability. No, viability is one issue. Another one is stability. So viability and stability need to be preserved. Okay, the stability preservation is a trick. Okay, for some bacteria they lose the traits very rapidly. Some organisms are quite stable. The typical plant associated organisms, they lose its stability. Okay, because they need that specific environment to retain its original traits, association. Okay, so these are all some interactions. So okay, what's going in the other group?
Criteria for a bacteria isolate which I am handling, to tell that it is that same species, we tell that 1400 best pair compare more than 90% of identity.
97%.
97% identity. Then what is the criteria for other organisms like fungi?
Yeah, fungi, the cutoff values are still evolving all the time, right? It's very difficult rather. So this question everybody is asking, and we are unable to come to a kind of consensus. 1400 base pair minimum length and 97% cutoff is suggestion by one group, right? And subsequent to that, lot of other suggestions have come. Why not the first 500 base or V3, V4? Lot of suggestions are coming. So my point is, if you depend on 16S ribosomal DNA, you go for the whole 1500 base pair. That technology is now slowly replacing. Okay, so one of the speakers highlighted. So cheaper technologies are available for whole 16S ribosomal DNA sequencing. Okay, in metagenomic work, instead of V3, V4, you go for 27F and 1492R.
Now we are realizing that read length is not enough.
Yeah.
Because 16 as amplified single sequencing, we are doing 1500 base pair. So the region is much long. But in V3, V4 in metagenomic data, we are having only 400 base pairs. Okay. So now we are thinking of doing the whole genome, this one, NGS, with the longer read length, so that we can able to cover the completely 16S, so that still it will give a more differences. Okay, we can expect more diversity we can get.
So more length is always better. So 16S full length is always better. But again, the cost will go up. Technologies are still evolving. V3, V4 is cheaper nowadays, around 12,000 per sample. So 1400 base pair means again cost will go up. It will be expensive also. It all depends how many samples you handle. Okay. So for diversity analysis, to get a kind of survey, this V3 V4 is fine.
Can get the overall idea.
They started a bacterial transformation practical and they is halfway through.
And this is the batch, isn't it?
Two batch they completed and another batch they are doing it now. And they will finally do the shocking here only. The machine we brought it here.
What about the group resolution?
We will have. It's already 5 o'clock. We'll put it in outgrowth.
What is this electroporation? What's this one? Sonicator.
Not sonicator, this is Electroporator.
Electroporator. It will hold single hold cuvette or two?
Single.
Single.
निकालना है या भाई पिछले सुर से चेंज नहीं करना है दिखा 4-5 बार दिखा दे
तो हो गया लग रहा है।
हाँ इसको तो दिखा चुके हैं 5-6 बार दिखाया है
Before this, no? So, what are other techniques are there? DGG, then another. TRFLP, okay. Done, next. Microarray, okay. So what is the advantage of DGG and the disadvantage of DGG? What is the limitations of DGG? The technique is same, only like what we are doing for NGS. We are isolating the community DNA, that is metagenome DNA. Then we are going for DGG. So what is the limitation of the DGG? Denaturing gradient gel electrophoresis. Not DGG. There also it's based on the length only. So how much we are amplifying for DGG with the DG clamp, GC clamp, how much we are amplifying? So there we are going to amplify only 250 or 300 base per only. So when we are seeing the tree length in NGS, we are doing 400 base pairs. Okay, little long, but that also not sufficient. Okay, so the initial technique with the DGGE, so still there are many people are doing, but the region for the comparison is still less there. Okay, only with the 250 base per or 300 base per only we are amplifying there. Okay, so when we are doing with the NGS with the 400 base V3 V4 region, even I am not happy even with that much only. Okay, as sir told that we can go for longer sequencing, that is a good technique, but other option is that we can go for TRFLP. In TRFLP we can amplify 950 base pairs. So there still your results data will give robust data. Okay, the comparison is still with the longer length 950 base pair. But only thing is, but still when people are going for NGS, because TRFLP it is somewhat laborious and time consuming. Okay, but the results what we will get is that it will be more accurate.
So now NGS of V3 and V4, you mean to say it covers entire 1500?
NGS of V3?
And V4.
No, V3 V4 is only 400 only.
No, others are now employing 16s. So in the training it is insisted that 16s of 1500 base pair it is advisable.
Yeah, that is a complete length of the 16s. Okay.
Otherwise,
See, many times what we are studying the single base pair variations. Okay. So when you are comparing the single base variations in 400 base pair and you are comparing single base variation in 1500 base pairs, so where you will get more differences means in 1500 you will get. So the your data which you are getting, there it will be more robust, more accurate in 1500 base pairs.
That is okay.
You are comparing with 1500 base pairs. Here you are comparing only with the 400 base pairs. Okay, and in DGGE you are comparing only with the 300 base pairs. Okay.
So that completes now whatever is in my this one research project, is it enough to go only for the V3 or V4 or?
See, in your project is with culturable one. So we are going for 1500 base pair.
1500.
And sequencing is based on Sanger sequencing only. Okay. In yours, only thing is will come is that whole genome sequencing part where we are trying to employ this genome assembly, these things and everything. Okay. Your initial characterization with the 1500 base pair is with the Sanger method.
Which we get only 800. 800 base pairs, no sir, out of Sanger.
In Sanger, in Sanger there are two sides you are going to do, na? So both forward and as well as in the reverse. Okay. In each side at least you are getting 700 base pair or 750 base pair.
1465 I think sir was telling it is optimal.
So that will come. Okay, you can able to form a contigs with 1400, 500 base pairs. You can able to form.
Primers that can be used for bacteria rather than this for amplification of bacteria.
See, for NGS one we are using V3 V4. Okay, even in this bacteria when you are comparing, this is for a eubacterial. So there is a V3 V4 is for eubacterial. Okay, there is a archaeal specific things are there. There also you can for metagenomics with specific with archaeal bacteria, archaea, it is available primers. So that you can make a separate library for archaeal based primers. Okay, so that also you can study. Not the domain specific. Domain specific. Okay. Eubacteria. No, they are not using for V3 V4. They are not using for. Still in this data we can get some one or two archaeal information also, but for making this one we can use specific to archaea.
If consider for 16 as whole length, can we get archaea also in that? Or?
Yeah, we can get in that. When we are increasing the read length for the NGS, that time we can get for archaeal this one also. Because our primers will be in such that it can cover both the domains. Okay, to amplify the 1500 base pairs. Cost it will be around 4 to 5. 4 to 5 lakh. In our division it is there. There, but it's not working.
Bring it here, bring it here. You can all round up here. Make a semicircle. Group wise. First group 1. 5 members should come here. Kuleshwar will demonstrate.
माइक लगाओ राजेश सर को लगाना है माइक
Sorry,
Put the mic also, explain. Other group can remain there in your place and you will call one by one. Micro beepers arrange.
Yes sir, this one.
Some can come that side.
Say it loudly and properly. Group 1, please come here with your sample.
So, till now we have make the competent cell.
Just before that. This is EQS, much like QS, but on both sides you have the electricity conductor, whatever you call it. Okay, so this allow the electricity heavy, that is 2.5 kilo volt to be passing through. Okay, and you will find a small gap in between that. There you can load your competent cells, which is mixed with donor plasmid and helper plasmid. Okay, so group number 1 and 2, they are putting 50 micrograms, 50 nanograms per microliter. 3 and 4, 100 nanograms per microliter. Group 5, mock. There is no plasmid. So give a shock. Explain.
So we have prepared the competent cells using 300 millimolar of sucrose solution, and then bacteria are it is ready with the plasmid. So I will now transfer these bacterial cells with the plasmid into the electrocuvettes.
Take the appropriate ones.
So there is a space where our bacterial suspension will go, and after that we will provide the electric shock.
You can give a gentle tap so that everything settles down.
Yes, sir.
So what he did is he just placed the 50 microliter of competent cell, which is mixed with the plasmids, in the small slot, right? And give a gentle tap so that everything settles down. Okay. And you see the proper orientation of your
yes, sir.
EQS.
So there is a small projection. So this will go inside.
So that you will be guided by that cartridge.
There is there is a small projection. So this should be, here is a same kind of like lock key model. So we will keep it like that and then insert it. Okay. After that, start button. Okay. Start button we have to put. Then we have to record how much millisecond. Now
some sound.
So small sound came, a beep sound, and now yes, it is done.
Take the reading.
Take the reading.
Time
it is 5.6 milliseconds.
5.6 milliseconds. So that's the time constant.
And
what
the
reading?
voltage applied is 2430
2430 voltage for 5 milliseconds.
5.6 milliseconds.
5.6 milliseconds. So if this only indicates that your electroporation was successful.
So now I will remove this.
Yeah, you remove it, put it in ice immediately. Just put it in ice, mark it, and go with the group number 2.
Yeah.
Now group number 2 people can forward and other people can the back side. Group 3, get ready there. You can move there. And group 4 also in line.
So this is the sample of group 2 having the 50 nanogram plasmid, both vector and donor, in the 100 microliter of the bacterial suspension. I'll first locate where is 2 number of 2. So yes. So I'll take the entire bacterial suspension.
Pay attention please, those who are standing behind, you can join the crowd. Let them be in the front You can be in the back, no problems.
So I'll put inside slowly. Yes. Oh, it is just ice. Nothing to worry. I'm putting the cap and then a gentle tap so that everything should settle down properly. Then just as I'm keeping everything in ice, so I'll wipe it so that there should not be any moisture outside. And again, using this projection, I will put it in the place of Electroporator. Then it is ready. Now I will start.
कौन सा बटन दबा रहे थे फिर एक बार
Start button.
अच्छा अच्छा वो तो पास से जाना पड़ेगा
ज़्यादा टाइम के लिए लगा पता नहीं. हाँ, it is 2390 volt and 654 millisecond. 654 इसलिए ज़्यादा आवाज़ आया. The time is little bit more. I don't know.
Got success?
Yes, sir. Some extra sound came this time. I don't know.
Yeah, what was the time constant?
654 milliseconds.
Okay.
Time is little bit higher here. 650 milliseconds.
Okay. I'll reserve my comments.
So any one of they will. So third group.
Go to third. It's always better to have both success and failure, so that you will understand which one is success and which one is failure. Ideally,
so
should not exceed 6 milliseconds.
Yes, sir. Earlier was that 5.6.
No way. Finished. Because salt concentration is a deciding factor. If it's too much salt, then this kind of problems will come. Group 3, please assemble. Other group can be in the back side.
Yeah, they assemble. So you want to do? कोई करना चाहेगा? So group 3. Here we have added. We have just double the concentration of plasmids. Here we are adding. We have added 100 nanogram. 100 nanogram both donor plasmid as well as the helper plasmid. So (100 microliter). I am loading entire content, that is 100 microliter. We have to load slowly so that it should go inside the gap properly.
You tap it well?
Yeah, yes, sir. I am tapping it.
Wipe it on the sides.
Center में ऐड है. It is, if you can see, it is in between the two electrodes. A very fine gap is there. There only. So
its maximum capacity is 100 microliter.
100 microliter. Its full capacity is only 100 microliter, and we have to fill the volume. So now we'll start the button.
So it is by default only, no? Then why it's showing again after ready?
No.
It is in that range.
No. The shock will be for very small time. Again it is.
Group 3, group 4. Yeah.
This time voltage is very less, 840 volt only.
Total 840.
Yes, 840 volt.
Time is small. So
We will discuss this result. Why this is happening.
So 3 samples we finished. Now group number 4, please come here. इसमें नाम नहीं लिखा हुआ. Okay, no problem. So the fourth sample I am adding now. Here also we have added 100 nanogram plasmid, both the vectors, donor and as well as the helper. नहीं कोई प्रॉब्लम, मैं थोड़ा बढ़ा दिया था. We have to put entire volume very slowly, otherwise it will go upside. Okay. So this is the projection which will guide us, like same lock and key models it is. So we have to put it in that direction only. So now it will go inside. So it is ready. Now I'll put the start button on. Why it is happening every time?
Proceed. Group number 5.
654 millisecond, 220 volt only.
220 volt. 654 milliseconds.
Why we are putting the E3 inside ice?
Now group 5 can come forward.
This is the control where we have not added any kind of plasmid. This control we are keeping so that we should know that after electroporation the cells are live or not. Whether it is and without plasmid, is there any other effect? Electroporation.
Just place it.
अब हाथ हटा लीजिएगा उसको लगा के.
Well inside. Okay. नहीं press.
लगा देगा और हाथ हटा लीजिएगा.
(2420 volt, 654 milliseconds.
Fine.
So
Done now?
it is done for all the samples.
Okay, what we have done now, each group was exposed to competent cell preparation. Then we divided the samples into 5 different groups. First 2 groups, 50 microliter was the sample volume, and we added 50 nanograms per microliter of plasmid DNA. You added 1 microliter, correct? That means 50 microliter of plasmid DNA, that is donor as well as helper, is mixed with 50 microliter of competent cells. Okay, then you gave a shock. The readings were recorded. That is voltage and time constant, and that is repeated with the other 4 groups also. Okay. Now the next step is you have to add the sock medium or LB broth. Right. Then you just incubate for 1 or 2 hours maximum. 1 hour also will do. Then after
1 hour, you can go for plating it on antibiotic amended medium. Okay. Now what you do, you just put it in the incubator, shaker incubator.
37.
If it is 37, for the bacteria you put 37. If it is 28, it all depends upon the target bacterial, the temperature requirement. Okay. We will do that, then you put it in the incubator, then we will break. Come to the auditorium, we will continue the 2 presentations. Come back and start doing the plating out. Okay, is that fine with all of you? Good. Now I request all of you to be moving to auditorium. Okay. Maybe 1 or 2 from, no, overall it's a simple exercise. What you have to do is you just add the media directly into this EQS, just mix it, right, then pipetting only, then put it in microfuge tube, alright, then allow them to shake for about 1 or 2 hours. Okay, after 2 hours just take it out and plate it on antibiotic amended LBA. Okay.